Thank you for having me here. Well, first time actually being around. It's okay. I forgive you not pronouncing my alma mater correctly because I didn't know how to pronounce your university. So I guess this is good. But next week you're gonna have another climate change and anthropology. So I guess I can sort of like try to lay little foundation of climate change before ahead of time for the next week's seminar speaker. So I'm a microbial ecologist, study of microbes in the environment, how they interact with others and stuff. And particularly the changing climate is one of the big factors for them to be responding and reacting. So let me ask you a question. When you hear about climate change, global warming, like the greenhouse gas, what do you have in your mind? What kind of stuff do you share?
The genocidal background of the people saying it.
All right. Wow, okay. Anyone else? What do you have in your mind when you talk about climate change, greenhouse gas?
Carbon footprint.
Carbon footprint? You've been watching your news, I guess. Yeah.
CO2 emissions.
CO2 emissions. And?
Heat and drought.
Heat and drought, okay.
Rising sea levels.
Rising sea level. Great, great. So I heard you guys are engineering, but sounds like very science people. Yes, they are all actually about the climate change, changing climate. Okay, because it is here, and it is actually happening right now, and we are trying to understand. Well, there's a number of different ways actually to see this problem, but see first, what can you do about this climate change? But another portion would be, what is going to happen when this thing is actually happening in 100 years? Let's say if CO2 level is, current CO2 level is about 400 ppm, and what if 100 years later, maybe 800 ppm? What's going to happen? What's going to happen? Do you care?
Yeah.
Do you care? And if CO2 is like all of a sudden 800 ppm, well, who cares? So if there's still plenty of oxygen to breathe, right? Temperature-wise, what if it's going to be like 3 degrees Celsius higher by the end of this century? Does it matter? So think about this summer. It was like this. Today is pretty nice out there, but this summer was pretty hot. How like 30 degrees Fahrenheit, at least higher than this. But you were still okay, right? 30 degrees was okay, but why we care about 3 degrees? Do we really care? Yes, we do, because, okay. Doesn't, come on. Which one?
Yeah, I got the little menu open.
Oh.
There we go. Now it's working.
All right.
There you go.
So just this Monday, a few days ago, Monday, Nature's Climate Change, which is the journal dedicated for climate change research, many different aspects of climate change, published this paper titled "Future Temperature in Southwest Asia Projected to Exceed a Threshold for Human Adaptability." What are we talking about here? Temperature changes, climate is changing, something happens to you, what would you do? If all of a sudden things been really cold or really hot, you might either move to other place, right? More pleasant place, or you would try to adapt to that new climate, maybe buying more coat, out boots and that kind of stuff, right? If you cannot afford those, maybe you have to die. Right? Oh my.
There you go.
Okay, so this is one of their prominent results from their paper, basically showing that this Saudi Arabia and Iran, that region, Gulf of Mexico, not Gulf of Mexico, that Gulf of Persia regions, that with this most pessimistic CO2 scenario, by the end of this century, it's going to be pretty inhabitable in terms of temperature, 60 Celsius, which is 140 Fahrenheit, if you didn't really get the number. Can you live there? Well, this is not Mars, right? Matt Damon was able to live inside of the... As a human in Earth, we're gonna go out and do some exercise and some stuff, outdoor stuff at the same time, indoor stuff. This is definitely inhabitable in terms of any physical activity outside. There are many factors contributing climate, changing climate we see. From many of those anthropogenic factors and some natural factors, most noticeably solar radiance. And all those things being calculated and estimated, then top three, CO2 and methane and nitrous oxides, are what's contributing to that radiative force of this climate change, this greenhouse effect. And because even if methane and nitrous oxide are a lot more potent in terms of per molar radiative force, but CO2 is so much abundant. They are most important and most problematic gas of this greenhouse effect. Anybody saw this before? This kind of graph? I mean, this is not artificial one. You might think it is artificial or something from engineering. It's not really new to engineering, but it's very uniform and very, systematic. It's almost a perfect oscillation each year. It is actually coming from natural system CO2 concentration level measured by at the Mauna Loa in Hawaii since 1958 to all the way up to 2013. It is continuously increasing. First time I saw was around this time, and it was ever since continuously increasing. And 2 years ago, May 7th, as a matter of fact, of 2013, for the first time in human history, the CO2 level reached the 400 ppm. And ever since, it again continuously going up. So we are actually having more longer period of time having more over 400 ppm CO2 concentration. And again, it just keeps increasing. Anybody guess why it had the oscillation, yearly cycle? Why summer is low and winter is high?
Lower plant life.
Lower plant life because why
Plants
lower plant
They go into hibernation in the winter. They slow down their
hibernation?
they slow down their processes.
What process are they talking about here? CO2.
Yeah, the photosynthesis.
Photosynthesis, that's right. Yeah, vitamin, good. So, CO2 uptake by plants are actually enormous during summertime, so they're actually going down, with the time going on. So if you measure pH of your local lake, pond, the pH of pond is actually being changed. It is higher. pH is higher in summertime and lower in wintertime, more acidic in wintertime because CO2 mixed with the water becomes carbonic acid and actually lower the pH in wintertime. So as CO2 concentration increases, it has a good correlation between global temperature increase as well as U.S. continental temperature increase and glacier volume decline, as well as the sea level increase. And also this CEI, Climate Extreme Index, which is one parameter, conglomerate all those different factors describing different climates of U.S. So the higher is more extreme. It's actually getting extreme and extreme as we see. If you remember, if you recognize the fellow's name, Jim Inhofe, from the state north of Texas, he is a leading voice. He's actually a ranking member in Environment and Social Work Subcommittee or something like that. He's one of those leading voices in government in climate change and environmental policies. He wrote this one, The Greatest Hoax, saying that it is part of the natural cycle. Yes, it has been natural cycle up and down for at least 800,000 years, but the recent trend we see in terms of CO2 concentration is way beyond that natural cycle range. And another group of people are also talking about solar radiation. Yes, there is, but it had the cycle of 11 years. And again, it doesn't really correspond that well with this cycle and the cycle we see, the pattern we see on Earth. So CO2 is increasing. And the model, there's a number of different models predicting what's going to happen. So this model was predicted in 2010, and we now have 5 years of data. and it looks like it's gonna be reaching up to those blue ones, much pessimistic scenario, at least for the last 5 years. And as CO2 concentration increases, and greenhouse gas and surface temperature, every surface temperature seems to be increasing. And again, looks like RCP 8.5 was the most pessimistic scenario is going to be the case we're going to have in the future. Okay, so then what's going to happen? IPCC, Intergovernmental Panel of Climate Change, a lot of people get together and talk about many different issues of climate change and wrote a report every once in a while. The last one was 2007, and latest one was last year, their assessment report 5. And at that time, this is what they are describing about North America. This decreasing snowpack in the western mountains, 5 to 20% increase in yield of rain-fed agriculture in some regions, increase the frequency, intensity, and duration of heat waves in cities that currently experience them. Sounds like something we see from the newspaper and mass media. So this is the updated one, and basically what they are updating here is that from 2007 to 2014 report is that they thought this might happen, okay, if the pattern continues in terms of CO2 emission and stuff. And 2014 report basically saying it is happening right now. Okay, that kind of stuff. One of the most obvious consequences we see, at least people, the Texans see, is the tropical disease pattern. Texas is not tropical. It is? Is it? At least this perspective, tropical disease perspective, it is actually tropical almost. Dengue fever is tropical disease by Aedes mosquito, but now it is, as you see, 28 states of the U.S. continent actually has a case of dengue fever, and you see in Texas, one of the most hard-hitting states. Waco, Longview is not actually free from those. That's not virus. It also was no case at all in 1999, and it's getting worse each and every year. And plant hardiness, if you ever, I don't know, engineering recognize this one, but when you grow something, try to plant some tree, like citrus tree, you need to have a certain temperature, lowest temperature that tree can withstand, right? So you cannot plant citrus in Minnesota and expect that's gonna survive and give you great citrus in summer. That doesn't happen because it's too cold for them to survive in winter time. So they actually, USDA update this hardness map every few years and rezoning basically those temperature zones. And as you see between 1990 and 2012, there is a definite shift upward. And this is those differences. So red, a lot more red zone than blue zone. So it's mostly temperature is increasing in U.S. continent. So there are a number of different reasons and evidences that there is climate being changed and shifted, and then we are actually facing the consequence of it. So as a organism, as a living organism, what would be the response of those changing climate? Again, I told you a little bit in the beginning that they can migrate, they can move around, they can move to different places, like those Aedes mosquitoes. They now, it is not that they don't like it, but they just have more territory for them to actually thrive. So they're actually moving upward, northward, and invading us, making us very sick. Or if they are plants, not just plants, but they are plants, they are normally cannot migrate, at least by themselves. So what they do is they adapt to the new environment, right? So adaptation can happen at their physiological level, called the acclimation, or it can undergo evolutionary response to change on their genetic level and genomic levels for the real adaptation. Or none of these strategies they can take, then they have to die, right? Like those potato in Mars killed all of a sudden because of the quick change of those temperature. they didn't have enough time to actually migrate or adapt to the lower temperature, whatever reason. So if it is that kind of situation, they have to die. And it results in loss of biodiversity. So we are very interested in the response of those climate change. So there are different groups of people actually studying what would happen in terms of CO2 and temperature-wise, so that they actually build models to predict them. And as a biologist, what we are interested in is as an organism, what's going to happen in 100 years later. We can still talk about them, we can still model them and see what's going to happen, but experiment, nothing can be experiment, right? So we got to build an experimental system to actually test that. How do you build those experiments simulating future scenario in terms of CO2 or temperature?
Closed.
We can
Closed system.
right. We can have a greenhouse kind of system and pumping in CO2, right? And increase the temperature at the same time. That has been the mainstream system, and some limitation goes to the closed system. It's not really exchanging freely with the environment. So early 90s, Brookhaven National Lab developed this technology called FACE, free atmospheric CO2 exchange enrichment technology. Basically, what's happening is this pipe and this facility, they're pumping out CO2 level they set. So again, we are, let's say, 380, 400 ppm range. If they want to simulate 600 ppm, for example, they pump out that concentration of CO2 continuously. So within this ring, inside of this ring, is locally maintained that enrich the CO2 concentration. And there are plants and there are microbes. So they are experiencing new environment, presumably future CO2 concentration. So you want to see what happens, right? It's a lot better way to actually than computer model or speculation. We can speculate a lot of things, but it is less accurate than this way. So the first study I'm going to present is what I did as a postdoc in work between Oak Ridge and University of Oklahoma, is that CO2 using one of those FACE facility up in the North Minnesota for the bioplant experiment. It had a biodiversity and CO2 and nitrogen treatment effect on it. So within this ring, there are different degree of plant diversity laid down, and we see at the same time CO2 effect on different diversity of those plants. Is there any factor? Are they doing anything in terms of their response? So you talk about photosynthesis. So we seems to care about CO2 concentration, 400 ppm to 800 ppm. Seems to be a big deal. Bad in negative way. How about plants? If you were plants, imagine yourself as a plant. Okay? Any your favorite flower or grass? You're gonna be happier with the higher CO2 concentration or less happier?
Happier.
Right, you should be happier with the more CO2 because
That's what you breathe.
right, that CO2 is their food basically to fix into organic material. That's the photosynthesis. Right? Anybody took biology, but there are light reaction and light independent reaction produce some ATP and NADPH, using them to reduce CO2 into glucose. But there is a way. So more CO2, they should be happier with the more CO2, right? And they uptake more CO2, then there might be no problem in terms of increasing CO2 concentration. How about microbes? How about microbes? When CO2 is happier, would microbes also happier? Why we care about microbes? Particularly in this case, soil microbes. When plants have more CO2 and they fix those CO2 into the organic material, what's going to happen to those organic material?
They're going to get broken down.
Well, they're gonna become bigger, the first thing, and they're gonna also use some of those to produce their own ATPs. So they're gonna become bigger, bigger and bigger. So your cow's gonna eat them, and you're gonna eat those cows, right? So there's something good about those stuff. They fix the carbon and they increase their biomass and basically support the ecosystem as a producer, right? But they're not like the most efficient machine in terms of using all those carbon they fix. It has been measured up to 20 to 40% of carbon they fix. It's been released through their roots to the soil for the root stage. So who are there in the soil? There are a lot of microbes in the soil, particularly the zone called the rhizosphere. There are a huge number of microbes there because of that fresh fixed carbon the plant just produced and released. Okay, so that actually is triggering factor for those microbes to be responding. Okay, and also, not only they produce those carbon substrate, they also produce other hormones and other so-called infochemical as a secondary metabolites. So they do actually have a very intricate relationship. In return, bacteria and archaea and fungi also produce other types of material, most noticeably antibiotics. Some antibiotics can actually kill off plant pathogens. They can actually help them to grow better. Some bacteria actually produce, mostly Pseudomonas produce plant growth promoting hormones. They actually help them to grow better. So there's layers and layers of different interactions between them. And one of the driving factors is carbon substrate they release at the root stage. So we were wondering if CO2 level is increased, plants gonna be happier, soil microbes gonna be happier, or just they don't care or whatever. So we want to see what's gonna happen in their functional level. Okay, what they do. There are many, many different types of microbes in the soil doing whatever activity you can imagine as a biological system, not engineering system, biological system. They can do. They can fix carbon, they can break down carbon, they can fix nitrogen, they can transform nitrogen from one form to the other forms. What would happen to those activities? So that's the treatment, and we looked at those microbes. So I'm a microbial ecologist, and I don't know, anybody took microbiology? No biology? No nothing? So I can lie everything else. So, again, biologists, let's say ecologists, who are interested in animals and plants and insects out there. So they go out, right? They study those systems. What do you think they would do as an ecologist? As a plant biologist, they go out, they look at them, they catalog them, they try to figure out what do they do. Okay, that's what we mostly doing, right? And I want to do the same thing with my microbes. Okay, how do I do that? Plants, you can go out, you can take a look at, right? You can count them, you can weigh them. Many, many different things you can do. How about microbes? Microbes are things, by definition, you engineering people, at least you should know about these. Things you cannot see with naked eyes, right? We are very limited in terms of techniques that we can use to study them. So again, what do you do as any experience with microbes? Microscope, very good.
Take soil samples.
Soil samples.
Soil samples to be able to.
Then what do? Take soil samples and do you ever do any culturing kind of stuff going on the media?
You measure something that they emit. So for pond water, you can measure how much gas they produce over a certain amount of time.
Right. So their activity can be measured, and you can go back to and estimate how much of those and what they really do. That's actually a very good way to study at the same time. But traditional way was like a Robert Koch, Louis Pasteur, those people, they were culturing. They're growing them so that we can actually see and we can actually study them. Okay? The problem is We cannot grow many of those. Actually, absolute majority of those we cannot grow. We don't know how to grow. Over 99% of those microbes in the environment, we don't know how to grow as of now. That's 99%. That's not the 1% we really know how to grow. So we had to come up with some other way to study using their DNAs and RNAs. Right? Again, engineering people, you should know what the DNA is, right? You all have DNAs. Every living organism has DNA, and their DNAs are similar in a way and also unique in other ways. So you can actually have something to compare each other. Right? So it's really good tool to actually study them, to look at how different organisms are out there, look at their genes, and we can actually estimate what they might be able to do. Okay, so this is the plots that we took sample. So this is called the 6S rRNA gene. rRNA, R represents ribosome. So ribosome is the organelle where protein is synthesized. And again, every living organism has to synthesize protein, right? Even if you eat your protein powder, still that's supplementary to what you're actually making it. So ribosome is really important. Every single organism has ribosome, meaning every single organism has a small subunit, either 16S or 18S RNA. So, and these are, they look like this. And there are some very conserved regions and very variable regions. So by comparing these sequences, we can actually compare human and your E. coli. Because there are a lot of portions we actually share between them. So we can tell how much different we are. Maybe dog, human, and E. coli. Right? That's fascinating, isn't it? So different regions of these 6Ns are supposed to be better delineating for different types of organisms. Some human microbiomes, usually we target B2 region, which is this region, and environmental microbes, which is those microbes we are usually targeting in my lab, is B4 region. Another technology is called the microarray. So particular microarray we manufactured in my postdoc lab was, so again, microbes have not only they have a 6Nes, but they have many functional genes to produce the proteins and enzymes so that they can do their activity. Let's say, if they are nitrogen-fixing bacteria, what I mean by that is, we know that what's the composition of atmosphere in terms of nitrogen? 78, 79, 80%, depends on who you are asking. So a lot of nitrogen, and we all need nitrogen, right? Do we?
Yeah.
For your protein, right? That's the reason that you actually take those protein powders, right? And nucleic acid. So we all need a nitrogen, but can we use nitrogen in the atmosphere? Why not? Why can't we use? Isn't it nice if we can just use them?
It's inert.
It's inert. It's very difficult to actually break them down and incorporate into organic biomass. So there is groups of organisms can actually do that job, called the nitrogen-fixing bacteria, most noticeably rhizobia in legume plants. You say legume is fixing nitrogen, but it's actually the bacteria living symbiotically living with the legume plants. So they fix nitrogen, okay? And so they have genes, dinitrogenase, the key genes to fix those nitrogen. So if you want to study those organisms, we can measure nitrogen fixing rate, amount of nitrogen it is fixed, actually ammonium to be fixed. Or we can measure those dinitrogenase genes, which is called NIPH, to study them, right? How many different types of organisms, or how much each of those different types are there under certain circumstances. So that is the basic idea behind this microarray technology, that we can test many different functional genes by one experiment, one hybridization, tagging this DNA or RNA, again, those raw material we're testing against, onto this array where each of these spots representing these different functional genes. So if there are a lot of those genes there, they're going to bind a lot more. So it's going to show up as a much darker color than the other. It's actually fascinating technology to study those cases. So a few data. So when there's more CO2, again, we talked about that more CO2, plants happier because they can fix more carbon. Yes, that's exactly what it is, right? Plants are growing, their volume is much higher, significantly higher under elevated CO2 than ambient CO2. How about microbes? They are responding similarly. They are also much higher under elevated CO2. Still makes sense because, again, 20 to 40% of their root X states, their freshly fixed carbon is released at the root X states. So they are given more food. They are going to growing better. They are going to have a much better, much more biomass. How about their composition change? Meaning, there is a community, members of different organisms forming community under ambient CO2, and there is a member of microbe forming another community under elevated CO2. Are they going to have a difference? Meaning, is CO2 change certain population of those microbes to grow better or grow less? Okay, and it looks like there is very clear patterns. This detrended canonical analysis is one of those alternation techniques to summarizing many different data points. In this case, we have, I think, 3,000 different data points simulated into three-dimensional data. And it's basically showing it is very nicely separated. I didn't talk about one. Another data. That data was actually from the sequencing, next generation sequencing technology, and it is quantitatively showing that yes, there is a very significant difference. Again, CO2 seems to have a very important effect not only on their biomass but also their compositional change. So some population is very rapidly responding, but not others. So we looked into their functional groups. Okay, so carbon fixation groups are these. Rubisco is the gene on the Calvin cycle. I don't want to go into detail, but these are genes on those carbon fixation. These are genes on the carbon degradation. Starch, your food, most of your food. Lignin, nobody eats lignin, right? Lignin is the major component of trees. We cannot digest lignin, but there are some bacteria that you can do. But what's happening here is that, yes, there are a lot more genes, functional genes, known to express to the enzyme to degrade starch and hemicellulose and cellulose than these more recalcitrant carbon. So they are readily responding to those fresh fixed carbon from plants. So these are a lot highly significantly enriched than the other. This data is really puzzling because most of their response was supposed to be through the plants, then the X states. But this one actually telling that they are also enriched carbon fixing populations. So groups of bacteria called the cyanobacteria can actually fix carbon. As a matter of fact, we think plants are the major oxygen producers. That's true only in terrestrial ecosystems. When you go into the ocean, it is microbes. Okay? It's microbes, cyanobacteria, and diatoms. So in global scale, plants may be half to 1/3 responsible for oxygen they produce. So Amazon is great, but so is cyanobacteria in the ocean. Yes, let's not, this is a little too much there. So let's not talk about that one, but still, so this is what we found out. Yes, microbes are responding very interestingly. Some of them is actually what we expected, some of them not really expected, but they were not like a skeptic, or they were like, didn't have an agenda, but they were just responding to the treatment of higher CO2 as through the plants as we expected. So that's the finding we had made out of this experiment. But I had a problem with this experiment. Even if I was part of that team and were able to actually publish in a pretty good journal. So this experiment was using phase facility, which was increasing CO2 concentration to 380 to 560 at one point. Okay, overnight, literally overnight, it just changed, and plants start to experiencing that new environment. Climate change doesn't happen that way, right? It gradually increasing as we saw those CO2 graphs, right? So, can we really trust the result we just talked about under that 1-point change data rather than gradually increasing data? There is a question there, right? This is a legitimate question to actually wonder there. The problem is, can we simulate gradually increasing climate patterns with the plants? We can, but we need 100 years to simulate 100 years, pretty much. How about microbes? If you can use microbes as a responding variables, if you can call them, we can actually shorten the time because microbes are very rapidly reproducing. E. coli, if you provide pretty good condition, they can reproduce every 20 minutes. Okay, we can start with 1, and in 24 hours, it can grow how many? 2 to the 72nd, which is more than you can ever imagine. It doesn't grow that much. Always there is a limitation. So it's actually rather good system to see rapid response. So my idea was between those abrupt change and gradual change, there's got to be some theoretical response difference. If it's quickly changing at once, they might not have enough time to adapt. If it's gradually changing, they might have a little more time to adapt. So that time difference, that difference might have some effect, probably most noticeably measured by community structure than functionality. That was my idea, and we actually had a little preliminary study between them to test them using cyanobacteria from the Lake Waco. Lake Waco is famous for cyanobacteria. It has a lot of them. Oftentimes they produce cyanotoxins, and it has been actually a persistent problem for many, many decades. That actually helped us to get funded from city of Waco. Doesn't mean that we didn't try to cure that problem, but it somehow it's persisting. It's persistently happening. We had 2 different chambers. One is quickly increasing CO2 level and temperature, and the other gradually increasing. Okay, and tested the response in the cyanobacteria using, in terms of their abundance and community composition, using 6-mass and their functional gene, microsystem producing gene called MCYD, the qPCR. So this is a profile that, yes, there is one chamber is one increase and almost maintained, the other is gradually increasing. Okay, so abundance. So we measured abundance in 2 different ways. Testing the gene abundance of their microsystem producing gene, MCYD, and chlorophyll A, which is measuring their chlorophyll concentration, which is also representing to the cyanobacteria concentration. In either cases, do you see any patterns there between gradually increasing and abrupt increasing? Some engineering people can see something, tell me, because I don't really see anything meaningful out of here. Yeah, chaotic. So in terms of abundance, we concluded there doesn't seem to be any huge difference. Let's skip over this thing. So this is multiple regression analysis. Try to determine the most important factor to determine this abundance difference. Even if their abundance doesn't seem to be very different between different treatment style, there is clearly both cases, both respondents, chlorophyll A concentration and MCYD contents, they are persistently choosing temperature and pH as a most important determining factors. So it tells me a little bit that there might be some difference in terms of what they are actually determining those abundance. Okay, and at the end, this is the one I was expecting that there might be community compositional level difference. What I mean by that is these yellow or gold if you put it that way, diamond is those gradually increasing chamber data, and this green circle is quickly increasing data. So overall, what you see is yellow is a lot more clustered, and green is everywhere basically. So my interpretation is, since the temperature and CO2 was quickly increased, they didn't have enough time to actually respond. Some of the population has to die for less abundant. So some other population is growing better and doing better than the other population. So community structure has been quickly changing. Yellow, since temperature and CO2 level is gradually changing, they might have better time to actually, more time to adjust, more time to adapt to the new environment. So their community composition doesn't necessarily be changing as drastic as the other one. So that's my selling point out of this experiment, and there is, yes, statistically very significant difference between these two treatments in terms of their clustering. So I was trying to selling this idea to NSF for 2 years now, because this is just two chambers, two data points basically, not two data points. There's no replicate. So at least this was already quite expensive. Actually, I had to spend quite a few dollars to set up this experiment and do all those analysis. So wanting to literally begging them to give me some money to actually expand the experiment, actually prove, or at least show that yes, there is a significant difference between two different schemes, then the consequence will be all the understanding we have based on the base study might need to be updated with this new CO2 or the temperature treatment. Instead of quickly increasing one time, gradually increasing will be the better way. We might need to actually modify what we are actually understanding. So they haven't actually, I haven't been able to convince them yet, but hopefully with the better wordings and better, more begging, I guess they might finally fund and expand this experiment and actually impact this entire field a little bit more drastic ways. So we are actually currently expanding this idea into solar system. Down in Temple, about 1 hour south of Waco, there is a USDA ALS station. They have this series of chambers, chamber from 250 to 500 ppm concentration. 200 ppm was 1850 CO2 concentration, which was, when was Industrial Revolution. Okay, and 500 ppm is probably late in this century. So we have a very nice continual gradient of CO2 from 1850 to maybe 2050 or 2070, 2080. And we have different types of plants there. One thing is we have switchgrass here. Switchgrass is one of those bioenergy crop selected and funded by Department of Energy. And we are actually proposing, studying their microbiome, how those soil microbiome affecting the growth and performance of the switchgrass from this changing CO2 gradient. And hopefully they like it, and we can continue to study and again impact this understanding of this climate change and microbial and plant response. With that, we'll take any question. Any questions? Sure.
So, to sum up what you're saying, because it's very hard for me to understand some of the things that you're saying, of course. But essentially, the way that we have been understanding climate change and measuring possible effects of climate change is possibly flawed, and that
I wouldn't say flawed, even if I think it's flawed. I would say it's limited. Indication might be limited. So we might, I mean, maybe I'm wrong. Maybe even quick change or gradual change might end up having similar effect. If that's the case, that's great. You can just continue doing, because it's going to be a lot easier for you to just quickly change in one time, right? So, I don't know. I'm expecting it's going to be different, but only like a bigger scale experimentation can actually prove it. That's my goal. I wouldn't say it's a goal.
Do you know if there's any other scientists or researchers doing similar research?
As far as I know, there are 4 similar types of research done and published, and all of them actually showing there's not exactly identical to what I'm proposing to do, but they were like stepwise change and like shortened time and that kind of stuff. But they actually showing some difference between 2 different treatments, like one-time change and some gradual types of changing. So that actually also giving more confidence that yes, there might be some difference. Yes.
So, would you say overall the effect that the CO2 increase had on the plants was beneficial?
So that's the ultimate question. So this entire question is about ecosystem response. So see, if we only have a plant in soil, has nothing to do, and we're adding a lot of CO2, yes, plants can actually grow a lot better. Okay? But what actually is happening is there's more CO2 and more higher temperature. So slightly increase the temperature is going to increase the enzymatic activity. So it actually helps them to fix more carbon much quickly. So photostatic rate is increasing rapidly in the beginning, but after a while they actually slow down. Okay, there are many explanations why they do that. And one of the factors is the microbial response. Microbes actually sucks in more, and they're actually changing the soil environment as those more carbon substrate is released into soil. That interaction we know a little bit, but we don't have a complete understanding. So one of the things we propose here is also having more complete understanding of studying those microbiomes. So you hear about human microbiome project, soil microbiome. So we are proposing to rhizosphere microbiome study as an entire system study. So that might be able to actually see better understanding rather than having piece of understanding, try to put together by speculation. Yes.
By using microbes and without plants, you can speed up the time scale, right? But are you worried at all that there's some interaction between the plants and microbes that might be missing when you take away the plants?
Exactly. So that the second experiment was called a microcosm, and microcosm. One of the drawbacks and criticism people make to the microcosm, it doesn't really simulate real situation, real ecosystem, because we just put in what we can control. Again, definitely, in ponds there is a fish, there is other small animals and plants and other stuff, but we don't simulate them because that's something we cannot really control and into the system. But yes, definitely there is some interaction, and that is the limitation that we had previously. So now we're actually updating to having better mesocosm, more realistic mesocosm, including different components from microbes to fish and plants and definitely in other aquatic organisms as well.
Any other questions before we vote?
Yes.
We will be.
There was
another
another question.
question.
What do you propose to do with your findings?
What am I put like a?
So let's say you find that there is a strong correlation or like between gradual and gradual CO2, like how it doesn't stress everything quite as much as everyone thinks. So what do you propose to do with that information?
So all policymaking IPCC report, and even like UN, there is a UN climate talk right now happening. They are based on, not I can't say all based on, but their facts on biological responses based on face study.
Okay.
So again, if there is a difference, and with more realistic gradual increasing pattern and the treatment, then we need to actually update that response. It might be more, it might be less drastic. Right? Depends on, if it's less drastic, then we might have a little more time. If it's more drastic, I think it's less drastic case, but if it's more drastic, then we need to, definitely hurry up to do something about it.
Yeah.
Right? Yes?
Does the increased abundance
of certain bacteria decrease the abundance of other bacteria in this?
So, yes, so competition, if those bacteria are in competing relationship, yes, they are. Such as they are targeting same substrate, but they somehow has different factor to affecting them. One population grow better than the other. Let's say if one is more tolerant to high temperature than the other, then everything is same. And if it's all of a sudden really warmer, then those more temperature tolerant population or thermophilic population is going to thrive. So that case, they can actually grow better by not as a result reduce the size of the population of the other ones, but as a result of the changing of the environment. But there also sometimes there is also direct competition between them by fighting of the same substrate. But again, it is actually as a community very complicated system. So we tend to actually study them as a response as a community instead of each population. There are just other people studying population-wise population response, but my lab and my interest is how they respond as a community. And what's happening inside, we sort of not look at really great in detail, but like see the response as a community is what we're actually looking at.
So when you took those gradient values, those were a community of bacteria, that wasn't just one species?
No, it is not. It is community of cyanobacteria what we are looking at.
All right, if you have more questions, Dr. Kang will be around here. Otherwise, we'll be going to the break room.